The ALPHA3 study, it was a trial in progress presentation. It's a study that we're all very excited about. By way of background, there's really two products being studied here. The first is an allogeneic CAR T-cell product called cema-cel. That is, it's an off-the-shelf CAR T-cell product that has a knockout for CD52 in the T-cell receptor. It targets CD19. The other product being tested is a circulating tumor DNA assay called Phase or Clarity. And we're studying these products in patients with diffuse large B-cell lymphoma. And so the way the study works is that patients are eligible if they have DLBCL that has gone into remission clinically by PET scan with conventional therapy. And they have to have sort of completed their conventional therapy, whether it's R-CHOP or Pola-R-CHP or whatever is given to try to cure their lymphoma. And they undergo at that time or soon after their end of treatment PET scan measurement of ctDNA in the blood. And if the ctDNA indicates that they have undetectable minimal residual disease or MRD, then they don't enroll on the treatment phase of the study, and they're considered to have an excellent prognosis because we know from prior studies that the large majority of the patients won't relapse. On the other hand, if they have an MRD test that indicates circulating tumor DNA and therefore residual disease, even though it's microscopic, they're enrolled on the study. And patients who enroll in the study are randomly assigned 1-to-1 between a control arm of very close observation where they get actually very frequent scans, or an investigational arm where they receive the cema-cel allogeneic CAR T-cell product after lymphodepletion with fludarabine and cyclophosphamide. So that's the treatment that the patients receive. Where there's two phases of the study, a part A and a part B, and we're conducting part A right now. And that's really to look at to do some safety and biomarker-based futility analyses. Once that's done, we'll then enroll a larger part B of the study where the primary endpoint is event-free survival. Again, testing whether this treatment modality can improve event-free survival compared with the more conventional approach of waiting until patients relapse and then delivering an autologous CAR T-cell therapy or whatever salvage therapy happens to be available at that time point. So that's the design of this ongoing study. It's enrolling at patients throughout different sites in the US and Canada.